Thanks to the organizers for inviting me here. It's been tremendous. So as Patrick said, my name is Kristian, and I'm from Department of Biomedicine at Aarhus University. And we are located here, right in the heart of Denmark. And a lot of the work that we do in our lab focuses on what we like to call genome-modifying enzymes. So this toolbox of enzymes consists of both the recombinases, for example, the Cre and P recombinases. We also have the DNA transposons, with which we have done a lot of work. We have the nucleases, so the zinc fingers, the talins, and of course the Cas9. So one of the major challenges when working with this toolbox is how we deliver it to our targets. So our target, it could either be in vitro in a cell, it could be in vivo in an animal model, or in the future it could maybe even be this little guy here. But if we break it down, there are basically three different ways we can do delivery. We can either do delivery by DNA, encoding our genome-modifying enzyme, either by plasmid DNA transfection or as viral genomes. We can also do delivery of in vitro transcribed mRNA. For both these methods, they, of course, both require intracellular production. And as Kieran also talked about, we can do delivery of recombinant proteins. And here, of course, we do not reproduce the protein outside the cells and then deliver it directly to our target. So one of the delivery platforms that we have been working a lot with is HIV-based lentiviral vectors. So very short, the way we produce these vectors is by a co-transfection of 4 different plasmids into virus-producing cells. So we have 3 different plasmids encoding proteins needed for viral assembly, and we have a 4th plasmid here, which is our transfer vector that will be packaged into the virus as RNA. Now, in this example here, the transfer vector contains expression cassette for the guide RNA and the SP-Cas9. So if we co-transfect these 4 plasmids into a HEC2-free T cells or virus-producing cells, after a couple of days of incubation, we can harvest the medium, and we can thereby do transfer of Cas9 and short guide RNA encoding genes into our targets. So, of course, after transduction, the transfer RNA will be reverse transcribed into DNA, which will then be integrated into the target cell's genome. Today's state-of-the-art lenti-based Cas9 systems is the lentiCRISPR system from the Gecko libraries. It comes in 2 different forms, but they basically contain the same elements. We have a U6 promoter driving expression of a short guide RNA, and then we have an EFS promoter driving expression of a Cas9, the SP-Cas9, fused to a puromycin selection gene to a peptide. So this of course allows us to do antibiotic selection for cells with successful integration and expression of our cassette. One of the ways we utilize the system is shown here. So here we have done a lentiCRISPR version 2 directed knockout of the CD20 gene in cancerous B cells, and we then look at the cell's resistance to treatment with rituximab. So rituximab is a monoclonal antibody recognizing CD20, so of course only cells that express CD20 will be susceptible to the treatment. So we did transduction of B cells, and then did puromycin selection for 14 days. When we did flow cytometry on these cells, we could see that we had a large population of cells no longer expressing CD20. When we took these cells and subjected them to treatment with rituximab, we could see that only the cells that had received a guide RNA targeting CD20 were able to survive. Another example is shown here. Again, we use the lentiCRISPR for transduction, but here we are interested in investigating how indels accumulate over time after transduction. So we did this both in HEC2-free cells and in primary dermal fibroblasts, and we basically see the same thing. That is that from 48 to 72 hours after transduction, we have the highest accumulation of indels. So we can use this information to estimate the optimal time for delivering a donor template for homology-directed repair. So as I said, characteristic for the lenti-based systems is that they integrate into the target cell's genome. And that's one of the things that we in our lab are trying to challenge. So we've asked ourselves, can we use these lentiviral particles as transporters of proteins instead of genes? So if we go back and look at how we produce these, as I said, we have 3 different plasmids encoding viral proteins needed for assembly. One of these is the GAG-POL. So if we zoom in on that, so the GAG-POL expression cassette produces 2 different polypeptides, the GAG and the GAG-POL. When they are expressed inside the virus-producing cells, they will assemble at the cell membrane together with our vector RNA. The virus will then bud off, after which the viral protease here will start cleaving the polypeptides, and we thereby have maturation of our virus. So our hypothesis is that we can take advantage of this mechanism and fuse proteins either to the N-terminus of the matrix here or to the C-terminus of the integrase. And we, of course, the vision for all of this is that we hope to be able to stuff viral vectors with the scissors needed for genomic editing. And thereby, of course, using HIV-1's intrinsic ability to deliver its cargo directly to the nucleus. So as I said in the beginning, we've previously done a lot of work with the DNA transposons, and that's also where we started with this project. So we began with the piggyBac DNA transposon, originally discovered in the cabbage lobber moth. So these DNA transposons are small parasitic elements. So all of they consist of is a transposase expression cassette flanked by 2 inverted repeats. And when this transposase is expressed, it will recognize the inverted repeats, cut the whole cassette out, and integrate it into another place in the genome. And it can do so either between genomic loci or from episome to genome. So first of all, we took the piggyBac transposase and fused it to the N-terminus of the GAG and GAG-POL polypeptides. And when we looked inside the particles that were produced by Western blotting, you could see that our piggyBac transposase variant here, a hyperactive one here, was effectively being packaged inside the virus and released. And we could see that this was depending on a protease cleavage site being present between the transposase and the matrix. So if we then look at enzyme activity in target cells after lentiviral protein transduction, for this we used our standard way of delivering transposons as control, which is a co-transfection of one plasmid encoding the DNA transposase, one plasmid carrying a transposon donor. So the donor we used has a PGK-pure cassette, so we can do antibiotic selection and quantify colony formation and use that as a measure of enzyme activity. So when we compared these two, our standard DNA transfection with delivery by lentiviral protein transduction, you could see that we had a significant increase in colony formation. So I know this is a very busy slide, and I apologize for that, but all you need to know is that what we wanted to do next was to, together with our piggyBac protein, to package the transposon donor needed for a successful transposition. So we produced lentiviral vectors with a piggyBac transposase fused to GAG-POL, and we had a transfer vector here, again with the, as before, with the PGK-pure cassette. Produced virus and harvested it, and did transduction in HeLa cells. And this gave rise to a high amount of puromycin-resistant colonies. And we could see that this was dependent on the transposase being active, as an inactive mutated form did not give rise to any colonies. But we could also see that it depended on the VSPG, which is the lentiviral pseudotype. And we also confirmed these observations in a range of other cell lines. So then we looked inside the cells, inside our target cells, after a delivery of our genome-modifying enzyme. And if we first take a look at after plasmid DNA transfection, what we saw were these cells that were absolutely stuffed with the transposase enzyme. We did this by confocal microscopy of HA-tag versions. Now this really shouldn't come as a surprise because we use quite a strong promoter to drive expression of piggyBac transposase. But when we looked at cells that had been transduced with our lentiviral particles carrying the transposase, we almost weren't able to see the transposase at all. And all we could see were these small foci here. So it appears as if that protein delivery is very restricted after a lentiviral protein transduction. So we wanted to investigate whether this was also visible over time. Oh yeah, sorry. So these two are comparable. They were done under conditions that were comparable. So you can basically compare these two images. Sorry. So we wanted to see whether the restricted presence of the enzyme also was visible over time. So the way we did this was in HeLa cells. And first of all, we did transposase delivery either by plasmid DNA transfection, by transfection of in vitro transcribed mRNA, or by lentiviral protein transduction. The cells were then incubated for a period of time before a transposon donor was delivered by plasmid DNA transfection. And as before, we used the PGK pure donor, which enabled us to quantify colony formation and use that as a measure of enzyme activity. So if we first take a look at how it looked after plasmid delivery. For the first days, we could see a massive amount of enzyme activity. And to our surprise, the activity lasted for almost 2 weeks before we were down to background levels of enzyme activity. And when we looked at in vitro transcribed mRNA, again, we had very high activity for the first couple of days, and as compared to the DNA transfection, the window of activity was shorter, but still the activity lasted. We were able to detect enzyme activity for nearly a week. If we then look at enzyme activity after protein transduction, again, at the first time point here, 4 hours after transduction, we have high amount of enzyme activity, and that it is comparable with the first time points from the other twos. But what we see here is that after 24 hours, we are not able to detect any active enzyme at all. So this essentially means that when we deliver our genome-modifying enzymes by DNA or RNA, enzyme activity over time is a matter of days, whereas it for lentiviral protein transduction only is a matter of hours. Now, so we are, of course, very interested in how can we use this system in different applications. And one of the ways that, one of the things that we are very interested in is seamless genome editing. So this was a technique for editing that was developed by Kosuke Yusa at the Sanger Institute in 2011, I think. So let's say that we have some genomic DNA up here, and we want to introduce a mutation at this point. We can do this by introducing a double-stranded break. After that, we supply our donor template. So this donor has, of course, it has homology arms. In one of the arms, we have the mutation that we want to introduce. And in the middle, in between the arms, we have a piggyBac transposon cassette. So this cassette contains a PGK promoter driving expression of a pure TK fusion protein. So the fusion part, the pure part, first of all, allows us to do positive selection for clones that have where homologous recombination has occurred. We can then screen the clones that we get for the right mutation, and when we have that, we can, because the TK part here, the TK is a suicide gene, so we can do negative selection when we remove the piggyBac transposon cassette. And what we end up with is essentially the exact same genomic DNA as we had to begin with, only that now we have our mutation of interest. And I think the beauty of this is that we can do selection all the way, so it's, yeah, I think that's very nice. And we of course hope that that we can use our lentiviral particles in the last step here, so to do genomic excision of these piggyBac cassettes by lentiviral protein transduction. So for this, we developed a new variant of the piggyBac transposase that is integration deficient but excision competent. So it contains 3 missense mutations, and so far we've tested it in a reporter cell line. So this cell line contains a CMV promoter driving expression of EGFP disrupted by the same piggyBac transposing cassette as you saw before. So first of all, we tested our new transposase variant by plasmid DNA transfection. When we transfect the cells, the transposing cassette will be excised, and we will have reconstitution of EGFP. Now this means that we can quantify the amount of EGFP positive cells and use that as a measure of excision activity inside from the genome. When we did this, we could see that our new excision-only variant outperformed the wild type significantly. So we moved on to package lentiviral particles with this variant, and again transduced our reporter cell lines with either the wild type or our excision-only variant. And once again, you could see that the excision-only variant was far superior compared to the wild type, both in HeLa clones and HEC clones. So now we're going to shift gears a little bit. We of course also want to deliver other genome modifying enzymes using this technique. And we just demonstrated that we are able to deliver both the protein and the donor needed for all-in-one delivery. So we of course hope to be able to deliver the scissors and the patch in all-in-one particles to do genomic surgery. We started this by looking at zinc finger proteins. So of course for the zinc finger proteins you need both the left and right finger. So we co-packaged lentiviral particles with the left and right fingers and did Western blot to look inside the particles, and we could see that both fingers were effectively being released by the protease, and this was depending on the protease being active, as a protease inhibitor did not yield the same band. We used a reporter cell line, first of all, to look at gene disruption after lentiviral protein transduction of zinc finger proteins. And when we did a surveyor assay, you could see that by increasing the amount of virus we transduced the cells with, so from 0 to 600 nanograms p24, we got an increase in indel formation. And we also confirmed this in 2 endogenous loci, the CCR5 and AVS1, in 3 different cell types: ectodermal-free cells, normal human dermal fibroblasts, and primary human keratinocytes, with indel rates ranging from 13 to 24%. So as for the, we of course also want to do all-in-one delivery. So we went back to our reporter cell line, which had a mutated form of EGFP, so a non-expressing EGFP variant. We co-packaged our zinc finger proteins together with the donor needed for repair. And as before, when we transduced the cells with increasing amount of virus as measured by P24, we could see that we got an increase in targeted gene repair. When we look inside these cells by confocal microscopy, we could see that for the first 12 hours after transduction, we had quite high amounts of proteins inside the cells, but 24 hours and 48 hours after transduction, we are not able to see anything at all. So this, of course, nicely reproduces what we saw with the piggyBac transposase. Again, we see that this, that the exposure to the protein is very restricted after lentiviral protein transduction. So we moved on to look at on-target, off-target ratios, and for this we looked at the CCR5 locus for on-target and the very similar CCR2 locus as an example of off-target locus. So when we did a delivery by lentiviral particles, we had on-target disruption rate of around 8%. For plasmid DNA transfection as control, we had around 7%. When we look at the CCR2 locus, that is for off-target gene disruption, whereas we for the plasmid DNA transfection do see indel formation at around 3%, we're not able to detect anything at all when doing lentiviral protein transduction. We have also done deep sequencing at several different targets and haven't been able to detect any off-target coding at all. So besides the EGFP donor, the mutated EGFP that we prepared before, we are of course also interested in delivering other donors by all-in-one protein transduction. For this, we turned to our all-time favorite, again, the PGK-pure cassette, and co-packaged this cassette flanked by homology arms together with zinc fingers. And these were transduced into the CCR5 locus on the AVS1 locus in induced pluripotent stem cells. First of all, we could see after delivery of our lentiviral particles, we saw a nice indel formation both from the CCR5 and the AVS1 locus. And when we subjected the cells to puromycin selection, we could see that transduction with our all-in-one particles gave rise to a high amount of pure resistant colonies as compared to a mock transduced iPS cells. We then isolated colonies both of the CCR5 and AVS1 locus and analyzed genomic DNA by PCR. We were able to confirm that we had site-directed gene insertion in 34 out of 38 iPSC clones. So with the zinc fingers, we are fortunate because they are quite small, and when we increase the size, as is the case for the tailings and also the SP-Cas9 and the SA-Cas9, we see a reduction in virus yield, but we also see an increased risk of internal cleavage. And we have so previous attempts from our lab to package the SA-Cas9 into lentiviral particles were unsuccessful because we had massive cleavage of the SA-Cas9 by the viral protease. Nevertheless, in collaboration with a group from Texas, we fused the larger SP-Cas9 to the N-terminus of the GAC polypeptides and packaged lentiviral particles with our new fusion construct. When we did a Western blot on the particle, we saw this nice fat band here corresponding to our Cas9 protein. So this was also being effectively packaged and released inside our lentiviral vector, our lentiviral particle. We tested this on primary T cells, and when we did delivery by lentiviral protein transduction, we got indel rates of around 15%. So I think a bit short, but in summary, the indel formation by lentiviral Cas9 shortcut and gene delivery is time and cell type dependent. Lentiviral particles can be adapted to deliver genome modifying enzymes. Nuclease delivery leads to targeted gene disruption in primary cells in up to 1/4 of the targeted alleles. You also see that endonuclease and donor for repair can be delivered together in all-in-one particles. Enzyme activity after lentiviral protein delivery is high, immediate, and short-lived. And finally, effective footprint-free excision is achieved by lentiviral delivery of excision-only DNA piggyBac transposase. And if you want to know more about all of this, we published a review last year in Human Gene Therapy. Of course, there's a lot of guys to acknowledge. HI, Sophia, Laura, and Emil, who did tremendous work on this. Jacob, our PI, who also think he did some kind of work. All the guys from Aarhus University and all our collaborators, and of course, thank you all for your attention.